In this video, we covering the unit 2 of the medical genetics. First of all, what is DNA? DNA is the deoxynucleic acids. Deoxynucleic acid means there are at two position. At the second position of the ribose sugar, there is deoxy, means there is no OH group. Okay. By the removal of this OH group, this ribose sugar is more stable as compared to RNA. In RNA, at 2' of the ribose sugar, OH group is present. So RNA is more reactive as compared to DNA. So DNA is a deoxyribonucleic acid, that is organic chemical of complex molecular structure that is formed in all prokaryotes and eukaryotic cells and in many viruses. DNA is the genetic material, right? What? DNA is the genetic material that code the information of our body. Which type of protein will be synthesized, what type of enzyme will be formed in our body, that all are decided by this DNA, that is our genetic material. And this DNA is inherited from one generation to next generation. That's why there is a transfer of the character from one generation to next generation with the help of this DNA, that is dinucleic acids, deoxyribonucleic acids, right? DNA is a double-stranded helical-like structure that is anti-parallel. If there are 3' to 5', the next is a 5' to 3'. DNA is a double-stranded helical form structure. See, this is a DNA. What? This is the DNA, that is double-stranded helical-like structure, and this is a nucleotide like A, T, G, C, and this nucleotide is combined or forming a bond in between this nucleotide with the help of hydrogen bond. What? Hydrogen bond. In AT, double hydrogen bond is present, and in GC, triple hydrogen bond is present. During the process of replication, what? During the process of replication, helicase is an enzyme. Helicase is an enzyme which break this hydrogen bond. And this both strand will be separate, right? This both strand will be form a helical-like structure with the help of this hydrogen bond. When this hydrogen bond will be break, they are separated with each other. So during the process of replication, we want to separate this helical structure, and this helical structure will be cleaved by helicase enzyme. After that, primer will be added. Primer will be formed by primase enzyme. After formation of primer, DNA polymerase and come to bind with this primer, and replication will be takes place in the direction of 5' to 3' direction. This is the process of the replication, by which new DNA strand will be synthesized. After replication, transcription will be takes place, and after transcription, translation will be takes place. So here is the chemical structure of the DNA. This is a sugar phosphate backbone. What? Sugar phosphate backbone. This is a ribose sugar. This is a ribose sugar, and this ribose sugar 1', 2', 3', 4', and this is 5', right? And this ribose sugar at 1 prime base will be attached. See, this base means nucleotide, nucleic acid, like adenine, guanine, cytosine, thymine. At 1 prime this base will be attached, and at 3 prime phosphodiester bond will be formed. What? Phosphodiester bond will be formed, and this phosphodiester will be formed and polymerized the sugar phosphate backbone. This phosphodiester bond will be formed by the 3 prime OH and 5 prime phosphate, right? And this will be takes place again and again, and this is the process of polymerization. DNA is a polymer made up of unit called as nucleotide. This unit is called as nucleotide. What? One unit is called as nucleotide, which contain nucleic acid, phosphate group and sugar. The nucleotides are made up of 3 different components, that is sugar group, phosphate group and a base. There are 4 different type of base, that is adenine, thymine, guanine and cytosine. This is the structure of adenine, thymine. Adenine and thymine is a what? Adenine and guanine. Adenine and guanine is a purine. There are 2 type of base, purine and pyrimidine. Purine having adenine and guanine, and pyrimidine having adenine, cytosine and thymine. This is pyrimidine. Pyrimidine having cytosine and thymine. And in RNA, this thymine is in place of thymine, uracil is present. What? Uracil is present. What holds DNA strand together? DNA strand are held together by a hydrogen bond between the adjacent base. This is a base, right? This is adenine, this is thymine. Hydrogen bond will be formed. Already I told. In AT, double hydrogen bond will be formed, and in GC, triple hydrogen bond will be formed, right? So breaking of GC, more energy will be required as compared to AT, right? Adenine always paired with the thymine, guanine always paired with cytosine. In DNA, in RNA, adenine paired with the uracil, because in RNA, thymine will be not present. From DNA to protein, the base on a single strand of DNA act as a codon. The later form 3 later codon, which form amino acids. What? The building block of protein. From DNA to RNA, this process is known as transcription, and from RNA to protein, this process is called as a translation, right? And the nucleotide that is contain or base that they contain, that are a code, that code for the amino acids, and that is a building block of protein. The enzyme RNA polymerase transcribe DNA into mRNA. It splits apart the 2 strands that form the double helix, then read a strand and copy the sequence of the nucleotide. The only difference between the RNA and the original DNA, that is the place of thymine, another base similar, that is the uracil. We use that uracil, already I told you. This is a sequence of the DNA, right? This is the sequence of the DNA, and after transcription, this DNA transcribed into mRNA. At the place of the thymine, uracil will be present, and these 3 codon code for 1 amino acids. That is a building block of protein, right? In multicellular organisms, the mRNA carries genetic code out of the cell nucleus to the cytoplasm. Protein synthesis takes place. Translation is the process of turning the mRNA code into protein. Molecules called ribosome carry out this process, building a protein from the amino acid coded form. Right? Protein are coded by the amino acids. That is building block of the protein. Right? Understand? So, next topic is karyotyping. What is the karyotyping? Karyotyping is the process of pairing and ordering of all the chromosomes of an organism. What? Karyotyping is the process of arranging or ordering all the chromosomes of an organism. Suppose human having 46 chromosome, 23 pair of the chromosome. What? Human having 23 pair of the chromosome, right? This 23 pair of the chromosome, 22 is the autosome and plus 1 pair is the sex chromosome, right? So, karyotyping is the arrangement. What? Arrangement of this chromosome according to the order. Means if they increasing the size of the chromosome according to increasing the size of the chromosome or decreasing the size of the chromosome. So arranging the chromosome according to the order, that process is known as karyotyping. Thus providing a genome-wide snapshot of an individual chromosome. Karyotypes are prepared using a standardized staining procedure that reveal characteristic structure feature for each chromosome. Actually, karyotyping, when we isolate the chromosome from an organism, we can see the chromosome under the microscope. Where? Under the microscope. When we see the chromosome under the microscope, how they look? We can identify the chromosome by the process after the process of staining method, right? There are different type of staining: silver staining, Giemsa staining. There are different type of staining. We can stain the chromosome and identify the process of karyotyping, right? Here is the process of karyotyping. See, this is a karyotypes. This is human male karyotype. This is human female karyotype. 22 pair of the chromosome will be same till 22 here and here both, right? And last is different in male and female is the sex chromosome. So here is the arrangement of the chromosome according to their decreasing the size, First number chromosome is the longest chromosome. Here also, after we see that there is a decreasing of the size in the chromosome takes place, right? So 46 plus XY, this is a male chromosome because XY containing male. Here 46 plus XX chromosome, that is female chromosome because XX will be containing a female. So if there any alteration in the chromosome or any changes will occur in the chromosome, that will be identified with the help of karyotyping. There is any aberration will be takes place in a chromosome, then that chromosome will be identified by the process of karyotyping, right? So karyotyping are very essential for process for the identification of the any abnormalities. Application. First, karyotyping help in gender identification, right? By seeing the chromosome under the microscope, we identify that this is a male chromosome or this is a female chromosome, right? If X chromosome are present, X chromosome are like this, Y chromosome are present, then this is a male chromosome, and if both X chromosome are present, then it is a female chromosome, right? So karyotyping help in identify the gender identification, right? Next, it is used to detect the chromosomal aberration like deletion, duplication, translocation, non-disjunction of the chromosome. So, where it is used? It is used to detect the chromosomal aberration. Means if there are deletion in any number of chromosomes, then you can easily identify. If there are duplication of any number of chromosomes, then we also easily identify by the deletion, duplication, translocation, non-disjunction of the chromosome. There is a process of the aberration. There are many types of aberration like trisomy, means there is a presence of extra chromosome in trisomy. There is translocation, with translocation piece of the chromosome, means here this is the chromosome, we translocate this piece of chromosome to the here. There are two types of translocation, periplasmic or paraplasmic, within the centromere or with the centromere. There are two types of translocation, right? Next is monosomy. This is the absence of one of the chromosome, like 22 pair of the chromosome plus X. Means there is absence of Y chromosome or X chromosome. This is known as monosomy. Deletion, there is a missing piece of the chromosome. If some piece of the chromosome will be missing, like this piece of the chromosome will be missing, then this process is known as the deletion, right? Next, it helps to identify the abnormalities of the chromosome like aneuploidy. It helps to identify the abnormalities of the chromosome like aneuploidy. By definition, aneuploidy cells have an abnormal number of chromosomes because each chromosome contains hundreds of genes. The addition or loss of even a single chromosome disrupts the existing equilibrium in cell, and in most cases it is not compatible with life. Actually, this all type of aberration, chromosomal aberration, will be identified during the fetus, during the formation of the fetus. We isolate the liquid from the fetal material and check the abnormalities if there are any abnormality as present in the chromosome or not. So this karyotyping are very helpful in identify the abnormality in the fetus. Right? Karyotyping are most helpful. That by two methods, this type of karyotyping will take place to identify the fetus. First, by the blood, by taking the blood of the pregnant woman, and by taking the amniotic fluid of the fetus. Next, it also used in predicting the evolutionary relationship between species, and genetic disease in human being can be detected by this technique. Genetic disease like Down syndrome, Turner syndrome, Klinefelter syndrome. This all syndrome are a genetic disease that caused by the aberration of the chromosome. In some, there is a addition of this X chromosome or deletion of the X chromosome, or addition of 21st number of chromosome, or like that. This type of aberration, chromosomal aberration, that is a genetic disease that are identified with the help of karyotyping. Next process is the chemiluminescence immunoassay. What is a chemiluminescence immunoassay? Actually, chemiluminescence immunoassay is a technique that are used to identify the immunochemical reactions, right? This technique illuminated the radiation. By that radiation, we see the immunochemical reaction. With the help of that radiation, we identify the immunochemical reaction present in a test tube, right? So, chemiluminescence is defined as the emission of electromagnetic radiation caused by a chemical reaction to produce light. Chemiluminescence assay is an assay that combines chemiluminescence technique with immunochemical reaction. In this chemiluminescence, chemical are used. That chemical, when react with immunochemical reaction, combined with that immunochemical reaction, they produced a radiation like horse peroxidase. When combined with this horse peroxidase, immunochemical reaction, they produce a light. This is known as chemiluminescence immunoassay techniques, right? This is the process of the chemiluminescence. In presence of HRP, horseradish peroxidase, okay, this HRP react with S2O2 luminol, okay, and this S2O2 luminol phenolate radical react. When they all react, oxidized luminol will form, and this oxidized luminol plus HV light produce HV light, and with the process of this HV light, we see the immunochemical reaction. This is antigen, this is antibody. This antibody contain the HRP. This is a chemiluminescence element, right? This HRP bind with this with chemiluminescence substrate. This is enzyme. This bind with this substrate, and after binding with this substrate, they produce a light. They are changing conformation of the enzyme, and they produce a light. Here another, this is antigen. This antigen bind with another a primary antibody. After that, they bind with secondary antibody. This secondary antibody contain enzyme. After substrate will come, chemiluminescence substrate will come and bind with this enzyme. Their conformation change will be occur. After changing the conformation, they produced a light. This is a chemiluminescence process. This process is the chemiluminescence. Application of the chemiluminescence. Application of the chemiluminescence. There are analytical application in analytical chemistry, in clinical chemistry, in clinical chemistry, identification of the drug, diagnostic tool, or like antibody, antigen. These all are identified in a clinical chemistry. In food chemistry also, and our study is focus on the clinical chemistry. We focus in a clinical chemistry because in medical genetics we focus in analytical chemistry or a clinical chemistry. So main application of the chemiluminescence is the identification of antigen, antibody, or identification of the drug or diagnostic tool in a medicine. Next is the genetic disorder. What is genetic disorder? The disorder that contain in a genetic or a gene, the disorder that takes place in a gene, or they are a disease which cause defect in a gene is known as genetic disorder. Sometimes there is a mutation. Sometimes there is a mutation, a change in a gene or gene. The mutation changes the gene's instruction for making a protein. So the protein does not work properly or is missing entirely. This can cause a medical condition called a genetic disorder. Suppose this is a gene, right? This is a gene. In this gene, alteration will be takes place. Alteration will takes place, and they cause a defective form of a gene. And after translation, defective protein will be formed, right? Then this process is known as mutation, that change or alter the whole process. So, in genetic disorder is a disorder that are present in a gene is known as genetic disorder. Types of genetic disorder. Actually, there are 3 types of genetic disorder. First is single gene disorder, second is chromosomal disorder, and third is complex disorder. In single gene disorder, mutation affect one gene, like sickle cell anemia, right? Like sickle cell anemia. Sickle cell anemia is a point mutation or frame shift mutation. Sickle cell anemia is a point mutation, means there is a change in a single base of the gene, or it is a frame shift mutation. After changing the single base, there are change in a frame shift. Whole frame will be disturbed. Right? Next is chromosomal disorder. In chromosomal disorder, where chromosomes are missing or changed. Chromosomes are the structure that hold our gene, right? Chromosome is a structure that hold our gene, right? If there changes or alteration will occur in that chromosome, then that act as a disorder. What? That act as a disorder. So that disorder is known as chromosomal disorder, right? Chromosomal disorder occur in a chromosome or the structure that hold our gene. That is known as chromosomal disorder. Example of chromosomal disorder is a Down syndrome, Turner syndrome, Klinefelter syndrome. These all are example of the chromosomal disorder. Next is complex disorder. Complex disorder are where there are a mutation in 2 or more genes. Means at a time mutation will take place in 2 or more genes and they form a complex with each other. That type of disorder is known as complex disorder. Often your lifestyle and environment also play a role in a complex disorder. Example of complex disorder is a colon cancer. What? Example of complex disorder is the colon cancer. So, first we discuss about the single gene disorder. What is a single gene disorder? Single gene disorder is nothing but it is a type of point mutation. In point mutation, what happens? There is an alteration of gene, either deletion of the gene, either substitution. In point mutation, what happens? Either addition will be takes place, deletion will be takes place, or their translocation will be takes place. Their missense mutation will be happen, nonsense mutation will be happen, right? This all are type of point mutation, right? in this type of point mutation, the example is sickle cell anemia. What happened in sickle cell anemia? This is our normal gene. This is our normal gene, right? GAG, that GAG, CTG. After transcription, they code for GAG, and this GAG after translation, they code for glutamine. Okay, this is a normal gene. Okay, normal gene. But in sickle cell anemia, after mutation, point mutation will takes place, and this A will change into T, right? This A change into T. After changing into the T, there is a U. What happened? U. And after transcription, at the place of T, U will be present, and this after translation, this GUG code for a amino acid, that is valine. What? That is valine, and this is a mutant protein. What? This is a mutant protein. What happened here in mutant protein? When this glutamine will change into a valine. When glutamine changes into a valine, the whole shape of the RBC will be changed into a sickle cell. The shape of the RBC is a biconcave, and they change into a sickle cell, and there is a deficiency in oxygen carrying capacity. What happened? Deficiency in oxygen carrying capacity, so that they called as a sickle cell anemia, right? So this is the process of the point mutation in sickle cell anemia that cause single gene disorder. Next is chromosomal disorder. What happened in chromosomal disorder? This is a karyotype, right? This is a karyotype of the chromosome. There are many examples of the chromosomal disorder, like Down syndrome, Klinefelter syndrome, Turner syndrome, and here we discussed about the Down syndrome. What happened in Down syndrome? Actually, in Down syndrome, it is a process of trisomy. Trisomy means presence of extra copy of the chromosome. Presence of extra copy of chromosome. This is known as trisomy, right? Result of an extra chromosome 21. Result of an extra chromosome 21. See here, this is 21, right? This is a 21, and this 21 number of the chromosome having 3 chromosomes. Actually, there are 2 chromosomes. Presence of a pair of chromosome, that is 2, but in this 3 chromosomes are present. That is a trisomy, right? Result of an extra chromosome, that is 21. It happened 1 in 700 children. 1 in 700 children. Characteristics is facial feature, short stature, heart defect, shorter lifespan. Has been controlled to the age of the mother, but can also be the result of the non-disjunction father's chromosome 21. This chromosomal disorder will be happen due to extra or more age happen in a woman. Pregnancy in late period. That time, what happened that time? during pregnancy after 40, there are non-disjunction will take place, or during meiosis, or during the separation of the chromosome in anaphase. There are process in anaphase where the separation of the chromosome, during separation of the chromosome, this will be happen, right? So this is the reason of the process why Down syndrome will be occur. Like Turner syndrome, in Turner syndrome, there is a loss of one X chromosome. Means 40 means there are 22 pair of the chromosome plus loss of one chromosome. In Klinefelter syndrome, 22 pair of the chromosome plus presence of one extra chromosome. These are the chromosomal disorder. Next is complex disorder. Complex disorder is the complex between disorder of the mixing between the different gene. So the example of the complex disorder is the colon cancer disorder. This is a normal gene function, right? Normal gene colonic epithelial cells, right? But in this what happened, BAT-R2 is present, MS1 is present, that cause a mutation. These both cause a complex mutation in the gene. After producing this complex mutation, there is no synthesis of the protein synthesis will be takes place, because phosphorylation will be not takes place here. Without phosphorylation, translation will be not takes place, and there is a formation of colon cancer. So this all are we discussed about the disorder that are present in chromosome, point mutation or complex disorder. Okay. So thank you.